Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I'm Naglaa Elmongy, professor of dermatology, Cairo University, and our video today will be about what's called keratinocyte neuro-immune units in psoriasis. The main reference I have used in this video is this nice review that was published in Frontiers in Medicine in June 2023, and its title is Keratinocytes Neuro-Immune Units, Collaborative Impact on the Initiation and Maintenance of Psoriasis. This is the second reference I have used also in this video, which is published in Frontiers in Immunology, October 2022, and its title is Soluble Mediators in the Function of the Epidermal Immune Neuro-Unit in the Skin. Here is the summary of the first reference. Psoriasis is a persistent inflammatory condition primarily caused by a specific inflammatory environment which is composed of units formed from keratinocytes, nerve cells, and immune cells, commonly referred to as keratinocytes neuro-immune cell units. And these keratinocytes neuro-immune cell units result from the interaction between stimulated epidermal keratinocytes, neural elements, immune cells, and the skin microbiota, forming a complex network of interactions. Many of these units come together to form interconnected and amplified loops, thus acting as a collective force to trigger and maintain the effects of psoriasis. Now I'm going to explain more deeply what came in this reference. The skin depends on various primary cellular networks to perceive and respond to environmental stressors like microbial dangers or physical and chemical irritants. Both the immune and nervous systems offer complementary approaches to defend the skin barrier, and the extent of which each system contributes to this comprehensive protective reaction of the skin is predominantly affected by the skin structures and time frame within which it can effectively operate. For example, the nervous system requires a time frame of milliseconds to communicate details regarding a barrier danger or threat from the skin to the brain. These signals typically originate at the terminals of sensory neurons within the skin and are then transmitted to the cell bodies of neurons situated in sensory ganglia, what's called dorsal root ganglia, before going to the spinal cord. From there, the signals travel to the brain where they are perceived as sensations like pain, itch, and other perceptions. On the other hand, the immune system functions within a time frame from minutes to days, predominantly using mobile leukocytes that travel to various areas of the skin to carry out protective functions. And importantly, we have to notice that epidermal keratinocytes alongside immune cells such as resident mast cells or dendritic cells within the skin are present in close proximity to axonal terminals, forming an anatomical arrangement that conduct regulatory pathways within the skin. And in this diagram, we can see how is keratinocytes and different immune cells like Langerhans cells and mast cells are in close proximity to the sensory nerve endings, forming the what's called keratinocyte neuro-immune cell units. So when epidermal cells identify foreign and danger signals, they will generate targeted immune reactions. The subsequent response of dendritic cells are also vital in terms of inflammation. Keratinocytes play a dual role, functioning not only as the responsible for the skin barrier, but also as active contributors to the innate immune system within the epidermis. In recent studies, the importance of keratinocytes has been identified as detectors of threats via the inflammasome mechanisms in psoriasis. Also, keratinocytes show numerous pattern recognition receptors and generate a large number of cytokines. Keratinocytes act as executors to actively participate in an immune micro network interconnected by cytokines. The epidermis has been considered as an initial phase in inflammatory processes, including the pathogenic interleukin 17 loop. I have already explained keratinocytes' role in psoriasis in another video, so you can watch it. Lately, neuroinflammation has gained recognition as a significant contributor to pathogenesis. Neuroimmune cell units represent anatomical units situated at the junction of the immune and nervous system, present in both healthy states and during disease conditions. Clinical investigations have established the key role of the peripheral nerves in the progression of psoriatic lesions. Administering botulinum toxin type A through injection proves to be an efficient therapy for individuals with psoriasis. Also, in mice induced skin inflammation using MEQ mode, inflammation was reduced through suppressing what's called TRPV1 positive sensory nerves. TRPV1 is abbreviation of the transient receptor potential cation channel subfamily V member. And actually, I will make a future video explaining details about this specific channel receptor. Also, in the same mouse model, inducing psoriatic itch was achieved by suppressing what's called TRPC4, or the transient receptor potential channel 4, expressed by sensory neurons. Also, after nerve removal, there is a reduction in interleukin 23 producing dendritic cells within the dermal layer during MEQ mode induced skin inflammation, leading to a clear decrease in interleukin 17 releasing gamma delta T cells. In the case of Candida albicans infection model, TRBV1 positive sensory nociceptors directly identify fungal agents and subsequently release calcitonin gene related peptide. Moreover, the senses of interleukin 23 by CD301b positive dendritic cells might be enhanced by calcitonin gene-related peptide, consequently stimulating dermal gamma delta T cells to produce interleukin 17A. These findings indicate that there is a potential interplay between keratinocytes and the neural systems in individuals with psoriasis. So the inflammatory responses occurring within the epidermis has a key role in immune reactions, and when combined with neuroimmune cell units, they form an integrated cycle to start psoriasis pathogenesis. The interplay among epithelial cells, the immune system, and nerve systems completes each inflammatory loop, forming the extensive circulatory inflammatory cycles in psoriasis. Cutaneous innervation plays a critical role within the peripheral nervous system. Epithelial neuroimmune cell units refer to specific anatomical sites where neuronal and immune cells can coincide and engage in functional interactions to regulate tissue pathology and physiological processes. Neuroimmune cell units have become as significant structures facilitating complex processes like inflammation, wound healing, and angiogenesis. Actually, in the epidermis, there is a co-localization of neuroimmune cell units. Certain studies have suggested that the proliferation of keratinocytes might stem from the cell-to-cell communication between keratinocytes and nerves, demonstrated by adding nerves into a human skin equivalent. Cutaneous nerve fibers interact with epidermal keratinocytes and immunocytes in the progression of psoriasis. Interactions between neurons and immune responses are mostly mediated by soluble factors such as neurotransmitters, neuropeptides, and cytokines. Also, keratinocytes have receptors such as neurotransmitters, acetylcholine, dopamine, and adrenaline, neuropeptides, and neurotrophins, which are closely related to the psychoneuroimmunological mechanisms. Cutaneous nerves consist of both sympathetic, and various sensory fibers. However, the specific functions of each fiber type remain uncertain. The skin serves as both a site for sensory interactions and a protective barrier, and it's innervated by sensory nerve. Uh, in addition, it is also supplied by a limited number of autonomic nerve fibers. Neurons create mesh-like clusters within the dermal layer with numerous nerve terminals extending into the epidermis. The dermal dendritic cells are in close contact to neurons, suggesting that neurons may regulate the functions of dendritic cells. To conclude, in the epidermis, the presence of keratinocyte neuro-immune cell units might play a role in creating an inflammatory environment where multiple of these units interact, thereby fostering an environment that sustains an inflammatory cycle in psoriasis. Immune cells have the capacity to express receptors for molecules originating from neuronal cells, allowing them to react to neuronal signals. On the other side, neurons also express receptors for immune-derived cytokines and neurotransmitters in a reciprocal manner. Immune cells and neurons could interact with keratinocytes in psoriasis. The epidermal factors, cytokine factors, immune cells, and nerve fiber receptors have the potential to shape cell neuroimmune cell units. These units create a complex feedback loop and cascade amplification important for psoriasis development. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.